It is my pleasure this afternoon to be telling you about the utilization of biomass, both lignin and the cellulose component, particularly with a focus on the lignin component and how that can be used to make materials. Obviously, in the cellulosic biorefinery, the biomass is pre-treated and there is a focus on the cellulose, taking cellulose to ethanol or other products. And in this pre-treatment, it is that removal of lignin that is so important to give access to the cellulose, and that lignin liquor, it becomes a waste. So if one really thinks carefully about that, that means in most biomass, lignocellulosic biomass, you're talking about 20 to 25% by weight being wasted. So it's very clear that to enable biofuels and to enable lignocellulosic usage, that lignin must be put to use for something. And of course, lignin is different depending on the biomass, whether you were talking about hardwood or softwood or grasses. And I left the representation for lignin from grasses out. But in general, besides being made out of aromatic components, as you can see in this diagram, we have these ether linkages shown in blue, whether you're talking about hardwood that has G and S components, or whether you're talking about softwood that's made only of G components. These ether linkages dominate in the lignin polymer. And the chemistry that we will focus on today is how can we make use of lignin in that pre-treatment in which we make valuable products from lignin up front. So that's again the concept that's been known in the literature as lignin first biorefinery. And if we're able to cleave these carbon-oxygen bonds and remove some of these oxygens, and as you could see from these figures, we can end up with phenolic molecules that could have very interesting value. So this is the idea of the lignin first biorefinery, or what's being known now in the literature as reductive catalytic fractionation. The reductive fractionation of lignin has been known for a long time, doing it catalytically and upfront while retaining a solid byproduct that's a delignified cellulose, allows us to continue with the second generation concept of the biorefinery, meaning that the cellulose can be used for making biofuels, for making ethanol, for making chemicals. But now up front, instead of that lignin liquor being a waste, we could convert lignin to these aromatic molecules, specifically these phenol molecules. For this presentation, I will focus on propylguaiacol and propylsyringol. We were very fortunate that we were among the very first groups to look at this reductive catalytic fractionation, and we published our early results in 2015. You could start with the lignocellulosic biomass and in a pressure autoclave reactor at 200 degrees, use different metal catalysts. I'm going to show you here the data for zinc palladium on carbon with methanol as a solvent. Since the wood biomass, for simplicity, let's say it's 25% lignin, then that means 12% of the starting biomass in total ends up in a liquid form and in these two monomeric phenol molecules. That corresponds to 50% yield in based on the lignin content. As I showed you a couple of slides or one slide before, the remaining solid is a cellulose that is still available to be used to make sugars from it and do other products. This process of catalytic fractionation or lignin first removal is really pretty agnostic. You get very similar results with different catalysts. You maybe get different selectivities of different monomers. We've published later on the use of nickel, for example, instead of the precious metal palladium, but it's very agnostic. It doesn't matter what biomass you use, poplar, birch, eucalyptus. You could even have miscanthus, We published on that as well as cornstarch. The excitement here in these phenolic molecules that we make is these can be used, as I'll show you in the later part of my presentation, as monomers to make interesting materials. But these are molecules now, and they can be used to derive other aromatic molecules that we already use. So they can be a source or a feedstock for drop-in chemicals that we already make and use from petrochemicals. And this is what this diagram illustrates. Once we make these two molecules from lignin, again, from wood biomass, that would be about 12% of the total biomass weight. The remaining ends up being a carbohydrate residue that can be used to make ethanol, for example. Notice further catalytic hydrodeoxygenation of this can take us to propylbenzene. Cracking of propylbenzene can take us to benzene. That's now one of the principal aromatic molecules that we use in the chemical industry. So one can make from benzene toluene, can make styrene by making ethyl benzene by the use of alkylation reactions from ethanol and benzene. Of course, paraxylene can be made from benzene as well as cumene and from cumene phenol. So notice this allows us to take the monomeric products that we made from lignin and enter as a drop-in chemical into a host of these different chemicals that are already being made and used in the petrochemical industry from petroleum. And the difference here is that we would be making these from lignin. That's one possibility. Now, before I go on, I have to explain, well, why this removal of lignin by using a catalyst all of a sudden is very successful versus a pretreatment with acid and base that leaves us a lignin liquor that is less useful. And the bottom line here is in acid or base treatment, in any kind of removal of lignin, you generate carbocations, and you generate those in the benzylic position. And these are very reactive with other phenolic group in the lignin. So what they generate in that liquor, often when we extract lignin, is a more recalcitrant material than the starting lignin. Instead of having carbon-oxygen ether bonds, now you have carbon-carbon bonds that are very difficult to break. And what happens in the catalytic fractionation of lignin is we use alcohol, such as methanol, for example, as a solvent. And that solvent plays a very critical role. It allows for the protection of these carbocations such that the recondensations are not happening very fast, giving the catalyst an opportunity to act on these solubilized fragments that have been protected with a methoxy group from methanol. And the catalyst now can deoxygenate these dimeric or oligomeric species and continue to break them down to the monomers. And we see very clear evidence for this methoxy group protection when we take a 2D NMR of the reaction solution. And we see a lot of the features that can be assigned to lignin, but we see this additional feature that we assign based on the two-dimensional carbon-13 proton NMR to this methoxy protecting group. So before I go now and talk about some of the uses of these phenolic compounds that we make from lignin to make materials, I want to show you that the cellulosic byproduct from this catalytic fractionation of lignin is very useful. It can be digested into sugars, and now we can use catalytic chemistry, and I'm illustrating here a couple of examples from our research group. Using ruthenium on carbon under hydrogen in water as a solvent allows you very quickly to reduce the sugar to sorbitol. And then in the presence of both ruthenium and this acidic polyoxotungstate catalyst at higher temperatures now, going to 240 degrees, again in water in the same solvent, we could take the sorbitol and make from it ethylene glycol and propylene glycol in very good yield. So that's another way to making chemicals from the cellulose byproduct. Alternatively, you could take the cellulose byproduct and digest it to the sugar. From an oxidation of the sugar, you make glucaric acid. And now by using catalysis in one pot, in one step, we developed this rhenium oxo platinum catalyst. And this paper has been published very recently, where the rhenium oxo is responsible for doing the oxidehydration, removing diol groups to these olefinic species. And then in the presence of a hydrogen donor, such as isopropanol, the platinum component of this catalyst does catalytic transfer hydrogenation, which means you make acetone from isopropanol and hydrogenate these double bonds to make adipic acid. Now, we know that adipic acid is used as a monomer in nylon. So this is now a drop-in nylon monomer that we could make from the cellulose byproduct from our catalytic fractionation of biomass. I mentioned that the phenol products, besides being able to introduce them as drop-in into aromatic chemicals that are used today from petrochemicals, these phenols are very valuable and they can be used to make thermoset. So there are two kinds of plastics, thermoplastics like polyethylene or polypropylene that have very large entanglement. And there are thermoset that we usually use phenol and bisphenol A, and we do cross-linking chemistry to make very rigid structural materials. I want to show you here with one of the products, this propylglycol or dihydroeugenol that I showed you we make from our palladium zinc catalysis from lignin. You could deprotect the methoxy group to make these this catechol, and by using epichlorohydrin, which by the way, epichlorohydrin is petroleum-based, but there's a company now in Thailand that actually makes epichlorohydrin from biomass. So epichlorohydrin can be bio-based, and you make this diepoxide. This diepoxide is very interesting because it's very similar to BPA. So all kinds of material that we make from bisphenol A, we can make from this epoxide by cross-linking with an amine. This is an example of a cross-linked thermoset that we made from this lignin phenol product. You could get really more creative actually with this. So for example, I showed you we could make this catechol from lignin. We can make from the sugars furfural and hydroxymethylfurfural. So now the idea is we deconstruct wood and then we bring these two components back together, the furfural from the sugars and the rigid aromatic structure from lignin, and we could make a multi-epoxide like this system. By bridging two of these with one of these, we make this structure, and by adding epichlorohydrin, now we have different branching points of the epoxide to make very interesting materials. Indeed, when we cure this tetraepoxide with amine, we make very rigid material. We make very rigid material, and I'm about to wrap up here. And we have a TG that's very competitive to what you make with bisphenol A. I hope I demonstrated to you today how you could start with biomass and with this reductive fractionation, make value from lignin up front. This can be used as a feedstock for drop-in aromatic chemicals or for making very interesting thermoset materials. The sugars can be recovered from the delignified cellulose and it can be used to make biofuels or make additional chemicals. I'll end by thanking the people that have done this work, the graduate students. Many of them have graduated recently from our group, and the Department of Energy, UCSB Champ Initiative, and a startup company that I founded that we collaborate with, Sparrow Renewables. And I thank you very much for your attention.
Hi everyone, I am Professor Luiziana Silva from the Laboratory of Bioproducts at the Institute of Biomedical Sciences, University of São Paulo, joining also the S2B Center at Innova USP. Thank you very much for the Biofuture and BBEST Organizing Committee for inviting me to talk to you about polymers and other bacterial bioproducts from biomass. There are a number of biopolymers, but we have been studying the polyhydroxyalkanoates, which are accumulated by some bacteria as granules inside the cell under specific culture conditions. These materials are a family of thermoplastics and biodegradable and also biocompatible biopolymers. And here we can see the general structure. And if it is a polyester, of course, and these are substituents can vary. A number of substituents at this point have been described in the literature, showing that the monomer composition can be highly variable. And this is very important because of its applications. PHA have a number of applications depending on monomer composition, starting, for instance, from commodities, as plastic bottles, films, going to agricultural applications, for instance, systems for controlled pesticide or fertilizers, deliver animal feed, and going to the medical applications as scaffolds for a number of use, stents, sutures, meshes, wound dressings, systems for controlled nutrient or drug delivery, hard patches, and so on. We can have a big list. And this is highly dependent on structure. If you take these main ester ligations as a backbone, and if you vary, these are substituent, for instance, with this CH3, we have C4 monomers as components of our polymer, which is then the P3HB polymer, polytrihydroxybutyrate, which has an elongation to break of 5%. And it is considered a short chain length monomer, as well the C5 monomers. If you combine C4 and C5 monomers, you have HBHV, and you can increase 10 times the elongation to break, improving material properties and application. And if you insert medium chain length monomers to the polymer, you have the medium chain PHAs, which are much more elastomeric, and they can elongate up to 1,000%. And interestingly is that bacteria either accumulate short or a medium chain length. And this combining different medium and short chain length is very difficult, and is a very promising area to be explored. And this ability to accumulate short or medium chain length depends highly on what you feed to bacteria, single or combined carbon sources. How do you feed bacteria? What are the culture conditions that you are applying to your cultivation? And of course, the genetic bacterial ability and metabolic pathways that give the appropriate intermediate to the PHA synthase, which is the enzyme that polymerizes the monomers into the PHA. What we have been doing is studying different carbon sources from biomass that can be used as raw material to produce these biopolyesters. We have been testing a number of sugars, first from sugar cane, but the literature reports a number of other biomass carbon sources. We have searched in the nature for different PHA producers, and we have selected some bacterial species, and mainly one particular new species, which is Burkholderia sakari, which have revealed to be a very good PHA accumulator. And more recently, we have been studying the use of hemicellulosic hydrolysates, mainly pentoses and cellulose. This strain has been very efficient with these first substrates. And also when we tested with sugarcane bagasse hydrolysate, we have seen that the efficiency in converting substrate to product, the rate of growth in this were the best described in the literature. However, the conversion of pentoses to 3-HB was low if we take into account the maximum theoretical use. And, of course, detoxification of the hydrolysate is needed. And, but the literature has been testing other hydrolysates with our bacteria very successfully. And to improve the ability of our bacteria, we have developed economic analysis, and we have concluded that some parameters based on actual results from experiments developed by Lopes and Schreiner, we have as a conclusion that we have to improve the maximum specific growth rate of the bacteria, and we have to reach 70% of cell dry biomass as PHA, and we also have to have productivity of this order. So we are developing different strategies to improve the xylose consumption. And firstly, based on the genome sequence, we have identified the operon encoding genes involved in the enzymes for the catabolism of xylose. We have also studied carbon catabolite repression and increased variations in the flux of some metabolic pathways. Also, evolutive engineering to the strain has been applied. And of course, given that you have this operon, you can enable different bacteria. You see that it's different from the E. coli operon, and you can enable a new bacteria to consume also xylose. For instance, if you go to carbon catabolite repression, we detected the classic that xylose only started to be consumed after the whole glucose have been disappeared from the medium, creating this typical two-phase growth curve. But recombinants strategically constructed led to a result that xylose and sucrose could be simultaneously consumed, and we had this very nice growth curve. And we also have observed that when you increase the expression of proteins involving in the catabolism of xylose, if you compare to the control, the biomass and polyester were highly improved when you overexpress these two proteins. And also you can see that the recombinant could consume faster the xylose than the control under bioreactor fed batch experiments. Thus, 25% was increased in the specific growth rates, 34% was increased in the polymer production, and yield increased from 0.26 to 0.35 grams of polymer per gram of xylose supplied. Also, to modulate the composition to vary monomer properties, we tested 30 different co-substrates. And we found out that C5, 4-HB, C6 were detected combined with 3-HB in the copolyester. Then we had 3-HB, 4-HB, 3-HB, 3-HX. And this C6 content in the copolymer can be controlled, varying glucose and hexanoic ratio. Hexanoic was one of those co-substrates. And as you can see here, we could improve up to 1.5 mole percent of C6 in the copolymer. However, only 2% of the hexanoic acid supplied could be converted to the C6 into the polymer. Thus, we combined molecular and bioprocess techniques to produce this copolymer, controlling the monomer composition, and that was this 1.5 maximum percent achieved. And then we could achieve up to 20 mole percent when we combined these two strategies. And also, when we varied the ratio of hexanoic acid and glucose, we could have different molar fractions of C6, and we reached up to 50% of the maximum theoretical yield, and C6 accumulation ability had a 25-fold increase. Also, to have a wide range and to control monomer composition, we have only by varying and applying specific bioreactor cultivation strategies, we could obtain a wide range of copolymers where the C6 fraction increased from 8 to 34%. And these materials are currently being evaluated as drug delivery systems for anti-tumor drugs. And interestingly, the cultivation was based uniquely on xylose and hexanoate as co-substrates. Basically, what we do is that if you go inside a bacterial cell, you can have a label, substrate, and trace where it's going in the product to see how are the fluxes, the metabolic flux working. For instance, you can observe that under non-optimized conditions, you have a specific metabolic flux for your substrate and co-substrates. However, if you define the theoretical maximum global yield, you can find out that you have to stop some routes to work, and you have to apply strategy to redirect the flux towards a new metabolism to promote maximum theoretical yields. You can also, inside your bacteria, insert new capabilities for production of new materials, and you can also apply different feeding strategies in bioreactor experiments. And by doing so, we have demonstrated that different bioproducts can be achieved by our bacteria using different biomass substrates. And basically, we want to have these bacteria working as bacterial factors, as mini factors, as bacterial chassis, and we can develop a number of other bioproducts. I have just told you about one of many we are developing in our laboratory, and ideally you could easily insert this bioproduction in a biorefinery, as the biorefinery of sugarcane and ethanol mill. And we are now joining the Synthet and System Biology Center at Innova USP, where we will be gathering with another group, different groups of experts towards innovation procedures using biomass. All of these results that we have been developing have been recently reviewed in this 3-volume handbook launched in 2020, and on it you will see that this production development in Brazil has been evaluated and considered a truly green process. And this is part of our team, our Brazilian team, and we have a number of other collaborations worldwide. And I hope you have enjoyed my presentation, and I feel very happy to have you in my laboratory, and I'm looking forward to your questions. Thank you very much for watching me.
Hello, everyone. First of all, I would like to thank the organizing committee for putting together this excellent conference. I'm very glad to be able to participate. My name is Mateus. I have been working for Braskem for the last 10 years. I have two roles at Braskem. One, I'm responsible for our research center in renewable chemicals. And the second one, that I have a transversal role supporting our business units with their renewable chemicals strategy. Today, I would like to share you a little bit about Braskem, what we are doing in renewable chemistry. I'm going to share some slides on our BioMEG project, and in the end, share Braskem vision on renewable chemicals and why do we think this is important to our planet. Just an introduction on the petrochemical value chain. You have usually the extraction, right? A company that is extracting natural gas, naphtha, and so on, right? So Braskem is consider itself integrated in the first and the second generation, which means that we buy naphtha or we buy gas, we crack those raw materials, and we produce the basic chemicals and some specialty chemicals as well. And we are also integrated on the second generation, in which we produce thermoplastics resins, right? So basically we produce polyethylene, polypropylene, PVC, EVA, and other materials as well. And then we have the converters, right? The companies that will produce the goods and that use plastics as raw materials. Braskem is a global company, right? We have industrial footprint in Brazil, Mexico, United States, and Germany. We have cracker activities in Brazil and in Mexico. In the US and Germany, we are mainly a polypropylene producer. It's a global company, right? We have presence in 27 countries. We have EBITDA of $1.5 billion, 80,000 people working across those countries, and we have invested something around $100 million in innovation on the past years. We have our main product today on the renewable chemicals are the polyethylene. So we built a 200,000 tons plant in the south of Brazil, in Triunfo, and an investment of $290 million. We produce several grades today, almost 40 grades of different polyethylene. But we also produce some chemicals from ethanol as well, right? Mainly ETB here, as mentioned in the slide. Recently, we launched with Alberts the green EVA, which is a polymer used on shoes, sole productions, especially. And basically, our main value proposition with Biopolymers, right, as you can see here, is the capacity of fixing CO2, right, to capture the carbon in the polymer, right? So from the photosynthesis, from the sugar cane that absorbs the CO2 from the air, producing the sugar, which is converted to ethanol. And further, this ethanol is dehydrated to ethylene and then polymerizing polyethylene. Basically, we are putting that CO2 in a very stable polymer, right? And this polymer will be used in several products, right? That can, especially when you're talking about long-term application, right, can capture this CO2 in construction, in cars, and other long lifetime applications, right? So basically, our polymer fixes 3 tons of CO2 per ton of polyethylene, and the conventional petrochemical polyethylene emits CO2. So for every ton of polyethylene that our clients use, they are abating 2 tons of emissions, right? When they substitute the petrochemical polyethylene, but they're also fixing 3 tons. They're taking out of the atmosphere 3 tons of CO2 using our green PE. So in a net, right, you have the reduction of almost 5 tons of CO2 per ton of polyethylene. We have a dedicated business in renewable chemicals, right? And we are very integrated in terms of innovation and R&D to the scale up and commercialization. So we have a team based in Cambridge, Massachusetts, focused on technology scouting, evaluating companies from Brazil, from the US, from Europe, from Asia that somehow are related with the chemicals business, chemicals and material business. We have an internal team working on pathway design to develop new pathways to new chemicals. We have a team focused on business development and market intelligence that are basically helping us doing our business case, market evaluation, trends, and so on. And then we have teams working on the development of the technology, right? From the lab scale in biotechnology, lab scale in catalysis, through application, product homologation, validation with clients, scale up until the delivery of the industrial plant and the production of those products. Just to comment one project that we have on our pipeline, right? We have a project to produce BioMG. And as you saw on the slides, to produce BioMG, you need to have oil refinery, production of naphtha, ethylene oxide, and then you produce the petrochemical BioMG. Our technology goes directly from sugar to BioMG. It's a very new, disruptive technology based on the cracking of the sugar. We have a partnership with a Danish company called Haldor Topsoe that we already built our demo plant in Denmark. We are working on the operation of the demo plant. This year, we are going to do the validation of the biomag, and we expect to have an industrial decision next year, beginning of next year. So it's a very interesting project, right? It's based on FCC, which is a very well-known project on the oil refining industry. So we are using the same concept to crack sugar into intermediates that will be hydrogenated to produce ethylene glycol and monopropylene glycol. So it's very flexible for different raw materials, from sucrose, from sugarcane, from dextrose, from corn, or even second generation sugars. So as you can see here, the reaction is you can get very high yields, right? So that's a concern here, and our expectation to have a project that will produce a renewable chemical in a very cost-effective way in comparison to the petrochemical process. So we are working today with the industrial location. So the first industrial plant location. So we are testing raw materials. We are understanding hydrogen availability. We are mapping consumers and also potential suppliers of raw materials. We are getting primary data on LCA of different mills or different sugars in different regions. And by the end of this year, hopefully we'll have a decision on the location of the plant. So as I mentioned before, right, it's a pipeline. It's a project that we have been moving forward on the demo plant, right? So this year we expect to have the not only the plant location but also the validation of the BioMG, the production of a bottle grades PET, and then having a plant location decision. Our vision related to biopolymers is that I mean, they can be an important tool for climate change, right? As I explained on the polyethylene, right? We can not only reduce the emissions for petrochemical polymers, right? But we also can capture CO2 from air and store in the polymers, in the biopolymers, right? When you see the total market for polyethylene, for polypropylene, and for bio-MEG, right, which are key materials, but I mean, we have dozens of potential materials that you could produce biopolymers, right? We get to a final number that it is hidden here on the slide deck, but it's almost 800,000 tons of a CO2 equivalent, right? So when we consider all the biomaterials and new applications from biomaterials, we can get to a potential reduction of more than 1 gigaton of CO2 emissions, right? Just to give you a reference, right? Globally, we emit around 50 gigatons of CO2, right? So can be a very relevant technology, especially if you consider negative emissions, that there are just a few technologies that can really deliver these negative emissions in goods. And we also believe that we are in a very good position, right? I mean, as you can see, we have been seeing, you know, carbon pledges, carbon neutrality pledges from countries, from companies, also in brand owners as well, which are very important to push the demand. We have been the advances of Paris Agreement. We have this ongoing discussion on the carbon market, but we are very positive. We are getting very good financial returns on the business as well. I mean, the tools in terms of biotechnology that they are evolving, so our capacity to deliver new technologies is increasing as well. We, as you can see, we have new products on the market. We have, you know, client demanding new products, new brands. And of course, in the end of this value chain, we have the consumers, right, pushing for new solutions and demanding new solutions. And we also have the financing instruments in place, the ESG funds being very aggressive in funding this type of companies, define this type of R&D as well, right? So when you consider all those aspects, we think we are in a tipping point in terms of sustainability. Past year, Braskem announced our carbon neutral pledge, right? So until by 2050, we not only want to become carbon neutral in our operation, but we also want to support our clients to become carbon neutral and support our value chain in the best way as possible. So that was my presentation. I'll be happy to answer our comments and questions. And thank you very much for everybody that is listening to this presentation and the organizing committee. Thank you.
Hello, my name is Giuliano Dragone. I am a senior researcher at the Department of Biotechnology and Biomedicine at the Technical University of Denmark. I will be presenting a study on the valorization of the cellulosic and hemicellulosic fraction of wheat straw through microbial production of lipids and carotenoids. Before starting talking about this presentation, I would like to introduce the research group where this study was performed. The group is called Biomass Conversion and Bioprocess Technology and is dedicated to develop efficient and sustainable process technologies for the production of chemicals, food ingredients, biofuels, and high-value compounds. We are located in the TU campus in Lyngby, north of Copenhagen. The work I am going to present aims to support the development of a bioprocess for the production of lipids and carotenoids by oleaginous yeast using both cellulosic and hemicellulosic fractions of biomass as feedstock for fermentation. The hypothesis behind this idea is that this strategy could lead to a sustainable pathway to produce these valuable compounds with potential to reduce production costs. To better understand how this work was performed, the study can be divided into three main parts. It will start with the selection of the best yeast candidate for utilization in hydrolysate. The second part is related to the adaptive laboratory evolution of the selected yeast to inhibitors derived from lignocellulosic biomass. And the last part deals with the development of an efficient downstream process in order to improve the extraction and separation of lipids and carotenoids from the selected yeast. So, in the first part of the experiment, 6 oleaginous yeast were initially cultivated in synthetic media containing glucose or xylose as carbon sources in order to identify the best lipid and carotenoid producer. As oleaginous yeast can switch their metabolism to lipid accumulation under nutrient-starving conditions, lipid and carotenoid production ability was measured and compared under different carbon-to-nitrogen ratios. The best candidates were then cultivated in cellulosic and hemicellulosic hydrolysates obtained from wheat straw, and their performances were compared. The results show that only Rhodosporidium toruloides, Cytoela coloradoensis, and Lipomyces starkii were able to metabolize both glucose and xylose as carbon sources for cell growth. Among them, Lipomyces starkii presented the best growth performance in terms of final cell density, reaching a cell mass concentration of 11.7 grams per liter in glucose media and 12.2 grams per liter in xylose media after 120 hours. Lipid and carotenoid production varied to the different yeast and carbon-to-nitrogen ratio. The highest lipid concentration, 3.6 grams per liter, was achieved by Lipomyces starkii cultivated in xylose media with a carbon-to-nitrogen ratio of 50. It was also found that the highest carotenoid concentration, 9.2 milligrams per liter, was obtained by Rhodosporidium toruloides when using xylose as carbon source and a carbon-to-nitrogen ratio of 50. This yeast also presents the highest carotenoid accumulation, almost 3 milligrams per gram in xylose media with a carbon-to-nitrogen ratio of 25. Based on the above, Lipomyces and Rhodosporidium were selected as the best producers of lipids and carotenoids respectively. This table lists the main fatty acid produced by the 6 oleaginous yeast in synthetic media with different carbon-to-nitrogen ratios. The major fatty acids produced by this yeast were palmitic, stearic, oleic, and linoleic acids, which accounted for more than 50% of the total fatty acid produced. The most abundant fatty acids produced by Rhodotorula glutenis, Lipomyces starkii, and Yarrovia lipolytica was oleic acid, followed by palmitic acid and linoleic acid. It is worthy to notice that the percentage of the oleic acid in Rhodosporidium toruloides increased as the carbon-to-nitrogen ratio increased. Fatty acid with 16 to 18 carbon atoms are optimal feedstocks for biodiesel production due to the similar fuel properties. The oleaginous yeast that produce the highest concentration of lipids and carotenoids in synthetic media were then cultivated in hemicellulosic and cellulosic hydrolysates at 30 degrees Celsius, 250 RPMs for 120 hours. Wheat straw was initially pretreated by hydrothermal pretreatment at 195 degrees Celsius for 45 minutes. The resulting solid fraction was hydrolyzed by enzymes at 50 degrees Celsius for 72 hours for production of the cellulosic hydrolysate. When Rhodosporidium and Lipomyces were cultivated in the cellulosic hydrolysate, both yeast metabolize all carbon sources in 72 hours. It is worth noting that xylose uptake started only after most of the glucose, more than 60%, was consumed. This sequential consumption of sugars by the oleaginous yeast could be attributed to glucose repression on xylose transport. The highest biomass concentration for Rhodosporidium toluloides was almost 18 grams per liter at 96 hours, while for Lipomyces starkei it was observed that the highest cell mass concentration was obtained at the end of the experiment, 120 hours. At 96 hours, the highest lipid and carotenoid concentration by Rhodosporidium were also obtained. When the hemicellulosic hydrolysate was used, both yeasts presented different fermentation performances. Glucose and acetic acid, for example, were completely metabolized by Rhodosporidium within 24 hours, while 96 hours were needed for the consumption of both carbon sources for Lipomyces. Furthermore, a long lag phase, 72 hours, was observed during the growth of Lipomyces starkei. A final comparison of the two strains revealed that, despite a sluggish onset of fermentation, cell mass and lipid productivity for Lipomyces were in general higher than those obtained by Rhodosporidium at the end of the experiment. In the second part of this study, a strategy of adaptive laboratory evolution in hydrolysate-based medium was developed to improve the tolerance of Rhodosporidium toluloides to inhibitor present in wheat straw hydrolysate. The ale experiment was performed in an automated liquid handling robot platform. Continuous fermentation of 10 replicates of the wild-type strain were performed in medium containing increasing concentration of wheat straw hydrolysate. To obtain different concentration of inhibitors for the experiments, the hydrolysate was diluted with synthetic medium composed by the same amount of sugars. The growth profile, lipid and carotenoid production of wild-type and evolved strains were then compared. The evolved strains present better performance to growth in hydrolysate medium with a significant reduction in their lag phases and improved ability to accumulate lipids and produce carotenoids when compared to the wild-type starting strain. In the best cases, the lag phase was reduced by 72 hours and resulted in lipid accumulation of around 28% and carotenoid production of 14 milligrams per gram in dry cell weight. We also observed that although the ability of the evolved strain to accumulate lipids was improved after ale, the evolution did not change the lipid synthesis pathway of the yeast. In the same way, the adaptive evolution did not change the profile of the carotenoids produced by the oleaginous yeast. In the last part of this study, we applied different strategies in order to improve the extraction and separation of lipids and carotenoids from Rhodosporidium toluloides. Saponification was used to fractionate the two bioproducts from the oleaginous yeast. The principle for separation of lipids from carotenoids is that in a sodium or potassium hydroxide-based solution, hydrogen ions of carboxyl groups or fatty acid can be neutralized and replaced by the metal ions to form fatty acid salts, which are soluble in water and ethanol, while carotenoids do not participate in this reaction due to the lack of carboxyl groups. Carotenoids can be separated from the fatty acid salts by extraction with suitable solvents. Meanwhile, saponification is a reversible reaction and fatty acid salts can be transformed to fatty acids again by acidification. We found that alcoholic potassium hydroxide saponification on disrupted cells or whole wet cells was more efficient to separate carotenoids and lipids than aqueous potassium hydroxide saponification. Purity of the recovered lipids was the same using whole or disrupted cells, but the purity of the carotenoids recovered was significantly higher when the process was applied on disrupted cells. We can conclude that it is possible to use cellulosic and hemicellulosic hydrolysates to produce lipids and carotenoids by oleaginous yeast. Among these yeasts, We found that Rhodosporidium tauruloides was the best candidate since it can produce both lipids and carotenoids. The adaptive laboratory evolution in hydrolysate-based medium was an effective strategy to obtain strains with improved ability to growth in medium containing biomass-derived inhibitory compounds. The process reported in this study allowed to extract lipids and carotenoids from oleaginous yeast with high efficiency and purity. Finally, I would like to thank Dr. Solange Mussatto and Dr. Xijia Liu, who were also part of the team involved in this research, and the financial support from the Novo Nordisk Funding and China Scholarship Council. Feel free to contact me if you have any question about this work. Thank you and have a nice day.
Hello everybody, my name is Solange Mussatto. I am professor in Bio-Based Technologies and Sustainability at the Technical University of Denmark, and today I will be talking about smart biomanufacturing, strategies to get maximum value from biomass. Before starting the presentation, I would like to introduce my research group. I'm head of the research group Biomass Conversion and Bioprocess Technology at the Technical University of Denmark. We are located in the main campus of the university in Copenhagen, is located in the north of Copenhagen, and at DTU we are at the Department of Bioengineering. And in the research of my group is especially dedicated to deliver innovative and environmentally friendly solutions in terms of process technologies, able to accelerate the transition to an economy less dependent on fossil resources. Basically, we work in all the steps of the biomass conversion chain. We start from the feedstock utilization. We apply pretreatment, hydrolysis to recover sugars and other valuable molecules from biomass. We also develop cell factories with enough robustness to work in medium produced from feedstocks. We develop the manufacturing processes. We are dedicated also to the use of statistical tools for optimization of fermentation conditions, for example, the downstream process for the product separation. And at the end, our purpose is to deliver new products, new technologies able to attend the society needs. In this presentation, I will be talking about smart biomanufacturing. And why is this important? If we want to move to a more sustainable and carbon neutral society, then the development of new technologies is crucial, because if we change the technologies that we have currently in the market for more sustainable alternatives, then we have also potential to reduce the emission of greenhouse gases to the atmosphere. We have the possibility to promote a more efficient utilization of the natural resources, including raw materials, minerals, and water. We have also the possibility to result in more cost-competitive production processes. And finally, if we develop something new, if we have new technologies, we have also the possibility here to create new job opportunities from the farmers to the small and big companies. The use of lignocellulosic biomass for new biomanufacturing processes has been strongly encouraged since these materials they are available in huge amounts worldwide, and they are also rich in sugars that can be used for fermentation. However, to have a fermentation process using lignocellulosic biomass as a feedstock is still, we have still several challenges here to overcome in order to deploy technologies with high efficiency for a large-scale implementation. One of the main points requiring improvement is the pretreatment step. So far, the technologies that have been used for pretreatment of lignocellulosic biomass, they have a high energy demand, which at the end results in a high cost for the overall biomass conversion chain. Selectivity is also not very high because it's quite difficult to release only sugars without touching in the other fractions of the biomass, including the lignin fraction. The efficiency of this process can also be improved, so that the main aim here is to recover as much as possible the sugars in an efficient way and without degrading the other fractions of the biomass. Then important efforts are still required here to take the maximum efficiency and the cost efficiency for the pretreatment step. The second point requiring attention is the fermentation, because fermentation using hydrolysate medium is not very easy. It's still not very easy. And the main reason here is the presence of different sugars in this medium. Then we do not have only glucose for fermentation, but we have a mixture of pentose and dextrose sugars. Not all the sugars are similarly consumed by the strains. Some strains do not have ability to consume pentose, for example. And apart from the sugar mixture, we have also several inhibitor compounds that are coming directly from the biomass structure, or they are formed during the degradation of sugars during the pretreatment step. At the end, we have here a very complex medium with different types of sugars, different types of inhibitor compounds, and basically the efficiency of the fermentation using this media is not comparable with the efficiency that we obtain when we use pure sugars as fermentation medium. Of course, the costs can be reduced when we produce medium directly from a feedstock. Then this is also the main aim, the main idea behind the utilization of lignocellulosic biomass. But fermentation is still a point requiring attention. From this part, I'm going to present two different strategies that we have been studying in my group in order to develop smart biomanufacturing. The first one, we have studied here the intensified pretreatment. And in this example, you can see we have used an intensified steam explosion pretreatment. In fact, all these assays were performed in a pre-pilot scale plant of steam explosion. And what we have done here was to change a bit the high torque limit of the equipment so that we could process more biomass at the same time. In fact, by doing these modifications, it was possible to process around 15% more biomass per unit of time. And also in this part of the screw, it was possible to increase to promote an effect of extrusion in the biomass, because we had more biomass entering in the reactor, then this also acts as an extra effect of pretreatment in the biomass material. We have applied this for two different biomass, for switchgrass and for eucalyptus. And we can see that in this table, the concentration of sugars and the main toxic compounds in the hydrolysate produced. In terms of sugars, what we could observe is that it was possible to increase the amount of sugars produced, but also a high amount of oligomers were present in the medium. Then in both cases, it was important to apply a post-hydrolysis process in order to have monomers, the sugars in the monomeric form, for using as fermentation. Also, what we could observe was, of course, a higher formation of inhibitor compounds. Especially when we performed the post-hydrolysis, we could see an increase in terms of acetic acid. The concentration was very high in both medium. This is also related to the higher amount of biomass processed, because acetic acid came from the structure of the material. Then, if we process more biomass, we can also release more acetic acid in the medium. And in fact, it was very high concentration here in both cases after post-hydrolysis. When we see in the table, basically, by intensifying the pretreatment, we got more sugars, but also more inhibitor compounds. This can also be a problem for fermentation, as discussed before. If we have more inhibitors, this media can be very complex and very difficult for use in fermentation. But then, if we think acetic acid and phenolic compounds, they also have some market value. Acetic acid has so many applications, and phenolic compounds as well can also be applied in different sectors, not only in food, but in pharma and cosmetic areas. So why do not recover these compounds? If we recover acetic acid and phenolics from the media, then we can add extra value in a biorefinery concept, and also we can favor the utilization of these hydrolysates for fermentation. In this case, we have then applied the detoxification application strategy where we covered both compounds, acetic acid and phenolics selectively, and with a very high efficiency, and by applying a sequential solid phase extraction in columns for phenolic recovery and the liquid-liquid extraction for acetic acid recovery. In the second strategy, also with the main aim of overcoming the inhibitor effect of the presence of the inhibitor compounds in the hydrolysate, we have been working on the improvement of the microbial strains. In microbial, we work very much with adaptive laboratory evolution, because when we have this complex media with the presence of so many compounds simultaneously, it's very difficult to apply genetic engineering, because the strain should be able to overcome the toxicity coming from different compounds at the same time. Then when we apply adaptive laboratory evolution, we can improve the performance of the strain to different compounds simultaneously. The Ale method can be performed in different ways, in sequential dilution in flasks, or using a chemostat, or even in robot platforms. The idea is to increase the tolerance of the strain step by step when we increase the concentration of toxic compounds in the medium. I have included here two examples. One is using a simulated hydrolysate medium. In this case, we have improved the performance of Chloromyces marxianus yeast with ability to produce xylitol and ethanol, but mainly xylitol. And in this case, we have elaborated a medium simulating the composition of the hydrolysates in terms of main inhibitor compounds and using xylose as main sugars. What we could see is that by applying a simulated hydrolysate medium, it was possible to increase the tolerance of the yeast to all these compounds simultaneously, and at the end, the concentration that we reached in terms of inhibitor compounds was very similar to the concentration that we find in the hydrolysates. We have done this for 4 different strains of Chloromyces marxianus, and all of them responded very well to the technology. In the second case, we have applied adaptive laboratory evolution using hydrolysate as cultivation medium. This is a more recent study where we have evolved a Rhodosporidium toluloides strain with ability to produce lipids and carotenoids, but also the strain is very sensitive to the concentration of inhibitors present in biomass hydrolysate. And in this study, we have used wheat straw hydrolysate as cultivation medium, and we have performed the experiments using a robot platform. The idea was the same, increasing the concentration of toxic compounds step by step. And at the end, what we could see is that not only the growth of the yeast was improved, but also the ability to accumulate lipids and carotenoids. In fact, adaptive laboratory evolution, we could conclude that is a very efficient method to obtain strains with better performance to grow in hydrolysates. All the strains that we have worked so far, they presented a better performance after Ale when compared to the wild type. When we perform the Ale using hydrolysate, it's even better than in simulated medium, because in simulated medium, we only mimic the concentration of some of the most important compounds. And in hydrolysate medium, we have even the compounds that are present in trace amounts, they are there, and the strain is also able to evolve to these compounds. Another interesting point here is that all these strains obtained by adaptive evolution, they are non-GMO, and they are also very interesting for application in food and feed industries. In terms of costs, if we have a robust strains, we avoid costs related to detoxification, and we can also improve the bioconversion efficiency from this medium. The only point that's still a challenge here is that since we evolve the strain to several compounds at the same time, it's quite difficult to determine or to identify from which compound the mutation is coming from. Then if we want to apply this information for reverse engineering, is still a challenging point. But in a bioprocess or biomanufacturing perspective, it's definitely very interesting. Finally, I would like to say thank you, especially for all my group members who have been working different talks as I have presented today. Also would like to thank from the financial support from the Novo Nordisk funding and the collaborators, especially from Uruguay and from the China Scholarship and Danish Technological Institute. Thank you all for your attention, and if you have any questions, feel free to contact me through the email here. Thank you.